Frontiers in Bioscience podcast series, where we discuss our past cosmic and biological evolution and our path to own biological and cosmic fate. I am your host, Dr. Sai.
Hello again. Thank you for coming back. This is the second part of what I call linear thinking or linear thought process. We went up, you look at the stars. Now it's time to go deeper into reality. Take a deep dive into what makes the microscopic world that is truly different and so bizarre than what we have conceived from celestial bodies and heavenly bodies. We, even from 5 BC, realized that if we take a bag and fill it with water, it can magnify what is underneath. But it took a lot of time for thinkers, the linear thinkers, to reach in 1620 to the development of the first microscope, creating an ocular and objective that allowed magnification of our tissues and cells. And imagine, even when I was a child, I was a curious kid. I asked my mother when I was 7 to buy me a microscope. And it was a beautiful day when I brought that small tiny microscope to my home. And the first thing I did was I put the wing of a fly underneath the microscope. It was blowing my mind. I could not understand the beauty that resides at microscopic level. And I'm sure in 1620, whoever is looking through this beautiful microscope is looking for the first time at something that mankind has never ever seen before. They started to examine everything in such a detail. Of course, there was no photography, so they started to make cartoons of these structures that were called cells. When they discovered cells, they thought they have reached to the indivisible components of life. But they were so wrong. There were people around that they started to think deeper. They wanted to understand, what is a cell? How does this work? How this bag of cells that we represent make us, make life possible. That was the intuition of people who wanted to look at the surface of these structures, and they created the scanning electron microscope. In fact, I was blessed that I was given the opportunity to work one of these beautiful microscopes, and I looked on the surface of these cells. It's amazing. It's filled with these microvilli, so much details that is truly unbelievable, that something that we cannot see, that has given us life, is so complex, nanostructures. And then there were those that they wanted to peer through these structures, to see the inside. They created the transmission electron microscope. Again, right at the beginning of my scientific endeavors, I was blessed to be allowed to go to the room where I could see these details. And I'm sure the happiness that I felt by looking at the cells under microscope would have been felt by those who would, for the first time, see the complex nature of these cells that make us who we are. The machinery that is inside the cells was complex. And it has taken a lot of pioneers, a lot of spirits, to understand how this beautiful machine works. There is a factory that makes energy. Another one that is the center where the proteins are made, the keys and the locks that makes the cell tick. It's not just simply structures that we now call organelles. It's bound and separated from the rest of the world around them by the by a membrane. These bags are separated from each other. But under no circumstances in our body, they are separate. They continuously have a dialogue. They talk to each other. They communicate. The celebration of life is the unselfish behavior of these cells. We wouldn't be here if we continued to be the selfish single cells that lived and were brought to this world 2.5 billion years ago. We don't have a fossil record of these beautiful creatures, the protocells, because there is no fossil record to see. We could imagine, we could just think how they could possibly come to life. But the question is, as biologists or as physicists, we think that the entire physics can explain the emergence of life by itself. There is no architect, no design of nothing. These atoms, they continuously bombard each other, and suddenly, oh voila, you have a cell. Well, we are going to talk about this in detail. It took so many years of work, consistent, persistent work in laboratories across the globe by so many scientists who dedicated their life to understand the cells. Now we understand the beauty that makes our cells work. The complex clockwork nature of the machines that is within our cells. They allow certain things to pass through this membrane. Certain things require these cargoes from outside to be delivered to inside, and the messages emanate everywhere. We have beautiful cables, actins and microtubules, on which beautiful structures, nanostructures that we are trying to build even today, they walk and they take their cargo to their destinations. We have learned about the complex signaling pathways, so many of them, that they work in concert to make life possible. There were other people who looked at the central headquarters of this cell. What is inside this nucleus of cell? Well, the two people who won Nobel Prize in 1953 by publishing their publication in Nature, there were Watson and Crick, who by X-ray diffraction reached to the conclusion that there is a code of life inside this nucleus, a double helix. This intuition, this insight, has given so many others to look for what is the meaning of this Bible that resides within every single cell that is alive. We reach to the conclusion that shows the beauty of life. The triple codon. Every triple code within this Bible of DNA accounts for one of the 20 proteinogenic amino acids that makes proteins that founds the cells and its structures, and it is important, without which we will not be alive. We understood how this whole code is transcribed from DNA to an mRNA, another structure that is beautifully transcribed from the code. This message is delivered from the nucleus to the cytoplasm. You have seen an egg. You have seen the nucleus. The egg yolk is where the nucleus is, where the DNA is, where the mRNA is. And then you look at the egg white. That is the cytosol. That's where all these proteins are being made, with one focus, to make this machine that apparently has no consciousness to work in a beautiful orchestrated fashion. If anything goes wrong, in this such a beautiful machine, everything stops. A cell that has been injured has to repair itself. And there's a beautiful DNA repair system that does this all the time on behalf of all of us, otherwise we wouldn't live. And then, when it really gets sick, when the DNA mutates, when the proteins get damaged, when the factory of energy stops, what will happen to this? Well, the cell has no option other than either commit suicide or become old, or goes through another process, metamorphose to cancer cells that they only understand they have to be in damage and they have to keep dividing forever. That kills the individual. The understanding of how our cells work was not sufficient for some other scientists. They have to understand, how is it possible that these molecules were born? How did they evolve? Where did they come from?